Hello, welcome back. Okay, so we're going to do an overview of the different structures involved in making a protein. So, quick reminder of what a protein is. Well, the building blocks of a protein are amino acids. And if you watch the video on how to draw an amino acid, it's N, C, C. Two H's come off the N. So this little area here is the amine group. And this is the next group, important part, the R group. So there's our amine group, the R group, and then the carboxylic acid group. There's our double bond oxygen and our hydroxyl. And this is, apart from the R bit here, the rest of it is all the same. Nothing changes there. So the only thing that does change is the R group. Now this table here, I got this from BioNinja. That's a really good resource to use as well. This is showing you how the different R groups. So the glycine has a really simple R group. It's just a H. That's what it is. Yeah. Whereas alanine, it's CH3. Let's make it a bit bigger so we can see a bit better. And then valine has got this CH with CH3, CH3. And then they're starting to get more complex. So like glutamine is a much bigger R group, CH2, CH2, NH2, double bond oxygen. So they're all differences. You've got arginine, massive R group. So because the R groups are different, the R groups have got different properties. So quite a lot of them are non-polar. So that would make them, they would, in terms of being in water, they're going to be hydrophobic. So they're going to orientate away from water. Whereas these polar ones, they're going to interact with water. So they're going to be hydrophilic. And then some R groups are just a completely negative charge. And then other ones are a positive charge. So in terms of bond forming, you can get ionic bonds forming when positive and negative charges come together. So there's going to be lots of different behaviors taking place based on the different R groups. All right. So another thing to remind ourselves is what a gene is. And a gene is a section of DNA and it codes for a particular protein. So the differences between different proteins are how those proteins are put together. And that information is coming from the sequence of nucleotides in your DNA. Okay, so the first stage of looking at protein structure, it's just, we call it the primary structure. What is the sequence of amino acids? So it could be something simple like, it could be only 2 amino acids that go glycine, alanine, glycine, alanine, glycine, alanine, over and over again, a big long chain of them. Or it could be that involves 5 or 6 different or more different amino acids. And all those amino acids are going to have all slightly different properties. So let's just quickly draw, really simple. There's an amino acid here. Join with another one, another one, another one. That's the primary structure. It's just asking what amino acids were put together and in what order did they get put together. Now this line here represents an important bond between the amino acids. We call that the peptide bonds. And that's the bond that forms, if you looked at the video drawing a dipeptide, it's the bond between the amine group and the carboxylic group. So those two hydroxyls interacting there's going to be a condensation reaction, and then you're going to end up with the 2 amino acids bonded together. So that's the primary structure. It's just asking what is the order of amino acids. Now that is really important because that's going to dictate everything afterwards. So the secondary structure, 2 different things can happen. When these amino acids start to join together, they can either, the chain can either begin to form a helix. and that helix is going to be held together by hydrogen bonds between the different amino acids. They're going to hold this together. So it's only hydrogen bonds formed here. Or another thing that can happen is so imagine there's two chains like that. If two chains can come into a line like this together, they can actually do something else. They can pleat like this. So there's one chain, and there's the other one. And they're going to get held together by, of course, hydrogen bonds. So far, we've talked about primary structure, the sequence of amino acids, and how it's held together by the peptide bonds, because that's how you stick amino acids together. And now we're talking about the interaction between different amino acids that can either form an alpha helix, this one here, or beta pleated. And those shapes are going to be held together by hydrogen bonds. Okay, then it gets a bit more complicated. So the primary structure has dictated the secondary structure. Now it's even more so dictating the tertiary structure, which is the 3D shape. All right, what actual 3D shape is going to be formed by this chain of amino acids? So if you look at a model shape of a protein, it tends to look like a big, like, folded up ribbon. You can take all these strange shapes, very delicate structure. So why is it doing those specific folds? It's the interaction of the R groups. So some R groups are hydrophobic. So if you imagine, you've got to think, proteins have been made in the cytoplasm, by the ribosomes. So they're surrounded by water. So you can see how the hydrophobic parts could end up orientating in towards the center here. Whereas the hydrophilic R groups, they're going to orientate themselves to these outer parts here, interacting with the water. So you've also got hydrogen bonds holding these weak little interactions here as well. And we've got ionic bonds. So if you remember, some R groups are negative charge and some are positive charge. So if you've got lysine, arginine, or histidine amino acids, they're going to be able to form ionic bonds with glutamic acid or aspartic acid. That's going to further strengthen and hold that shape. So imagine if this amino acid here has a positive charge, because the R group's got a positive charge, and this one's got a negative charge. Well, you can see how this could start to fold over. And then that negatively charged amino acid is going to start to interact with that one. So you've got this new chain starting to fold over onto itself. Okay, so this is just showing how this, or whatever, yeah. So I'm not drawing that very well, but how it's that the negatives and the positives are attracting. And you can imagine if these two here if that was negative and that was negative, they're gonna be repelled from each other. They're gonna move away from each other. So these different interactions based on their properties there. So that's the ionic. So there's we talked about hydrophobic, hydrophilic, ionic. And then you can get disulfide bonds, and these are the strongest bonds that you find in the tertiary structure. And disulfide is only going to happen with amino acids whose R group has got a sulfur atom. So let's have a look. We've got one there. So cysteine has got the little S there. Let's see what others we've got now. You've probably seen more than me already. There we go, another one, methionine. There's more, just not looking very well. So if they've got these sulfurs, then they can form bonds with each other, called disulfide bonds. Okay. Right. So once that happens, you're going to get two very different types of tertiary structure. One is globular, which tends to be hormones and enzymes, and the other one would be your more structural proteins, which are fibrous, like keratin, which is in your hair and nails. And then the last level of structure now. So that's quite a lot of bonds there to remember for the tertiary one. There's a lot to remember for this one. Finally is what we call the quaternary structure, and it's asking, okay, so you've got your primaries, it dictated the secondary, and it's dictated the tertiary structure. Now, how big is the actual protein? If it's just made from one polypeptide chain, this is a polypeptide chain that's folding up on itself, then it doesn't have a quaternary structure because it's just by itself. If, however, another polypeptide chain starts to fold within it as well, we say now that it's got a quaternary structure because it's got two or more polypeptides joined together. So hemoglobin is a protein in your red blood cells, and it contains 4 polypeptide chains. It's got 2 times alpha helix, and it's got 2 times beta pleated. So 4 different polypeptides working together to make one big hemoglobin protein. And it's also got something else. So another thing that will make, it doesn't always have to be, but it's something else it can have. It has what's called a prosthetic group, which is a non-protein structure embedded within the protein, which hemoglobin has as well. It has something called heme groups. So they're these non-protein structures within hemoglobin. There's 4 of them. And they contain iron, the ion, which allows it to bind on with oxygen much easier. That's what hemoglobin's job is to carry oxygen around the body. Okay, so 4 different structures. You need to know what the overall initial part of it is. So primary is just the sequence, and you need to know the bonds, peptide. Secondary, there's only 2 shapes, alpha and beta. The only bond you talk about is hydrogen. Tertiary gets more difficult because it's a 3D structure now. We've got to talk more about the interaction of R groups. So we've got hydrophobic, orientating away from water, hydrophilic interacting with water. We've still got hydrogen bonds happening, ionic between the different charges of R groups, disulfide only between those R groups which have sulfur. And the 2 shapes you get from tertiary are either globular or fibrous, and then quaternary has to be 2 or more polypeptides. For example, hemoglobin has 4, and it can also contain a prosthetic group, which is a non-protein structure, which hemoglobin has as well, which we call the heme group. It's a non-protein structure. All right, see you later.